All right, if you are watching this and listening to this prior to the first day of school back after the winter holidays, then this is out of sequence in the notes packet. So you need to flip to and find where it talks about the scientific discoveries toward the structure of DNA. That's what this is covering. And my phone is sitting there just because I wrote something on this, and then I needed to restart the video based on something that I said. So anyway, that being said, here we go. Frederick Griffith in the 1920s, he came up with this idea of transformation. And notice this says perhaps right here. So he wasn't totally sure as to what was going on with what he discovered with the graphic that we're going to talk about down here. So he did some studies where he used bacteria that was pathogenic or virulent and could kill mice, and then some that was non-virulent or non-pathogenic. So he discovered, and he said perhaps one form of bacteria could transform another form of bacteria and cause it to be virulent or pathogenic and kill the mouse. And he said maybe this was because of what was known as a gene that Gregor Mendel had named in the mid-1800s. So what Griffith did, just to, like I said, kind of keep this short and sweet, is that he had a virulent or pathogenic strain of bacteria that he injected into mice, and those mice died. And he did the same thing, injected bacteria into mice, but that bacteria in the case that's shown here, that bacteria was non-virulent or non-pathogenic, so the mice lived. Then he heat killed the virulent strain, and so because that bacteria was now dead, he injected it into mice and they survived. They lived too. Then he mixed the living R strain, which is the blue non-virulent or non-pathogenic strain, and then the dead S strain, the pathogenic or virulent one, injected that mixture into mice, and the mice died. So he said maybe one of these bacteria was transformed into being virulent by the other one, maybe because of a gene. Didn't really know for sure, of course. So then we've got Oswald Avery in 1944. He piggybacked on Frederick Griffith's studies, and he used enzymes to destroy organic compounds. He basically, so just think of this to keep it simple. He had enzymes that would destroy carbohydrates, lipids, proteins, and nucleic acids, or 4 types of macromolecules, okay? So after destroying DNA, specifically the transformation that Griffith studied, that did not take place. So Avery said that that is probably what was the genetic information, the DNA was, okay? And again, that he did this breaking down different types of macromolecules using enzymes, which we've already talked about, okay? So make sure you do remember the function of enzymes. We're going to keep talking about them all year long. Then in the early 1950s, Hershey and Chase, they used radioactively labeled bacteriophages, which that's a bacteriophage right there. And that little pink circle that I drew, just imagine that as a bacterial cell, okay? And so if you remember from the term phagocytosis, that PHAG means to eat or to feed. Okay, sorry, that weird sound as I drug my phone off of there. So these are specifically viral particles that look like little lunar landing modules that infect bacterial cells only. Okay, so they are bacteria eaters is where their name comes from. So that's a little viral particle. So Hershey and Chase labeled DNA with radioactive phosphorus and labeled protein with radioactive sulfur, which I just realized I don't have an S for sulfur. Okay, so its chemical symbol is just a capital S. So the DNA that you can see up here in the top portion of the bacteriophage, it gets injected down the height or length of that into, like I said, this is a bacterial cell. So they then studied the bacterial cells, and they were found to have radioactive phosphorus after the bacteriophage was attached to the cell. Therefore, the genetic material is DNA. That was kind of a very solidifying, or was, I shouldn't say kind of, that was a very solidifying experiment that they did, or series of experiments. Oh, and there was no radioactive sulfur in the bacterial cells. Again, that's my little quick draw of one. No radioactive sulfur in those when they studied the bacterial cells after attaching the bacteriophages. All right, and then truly from a structure standpoint, not just that perhaps DNA is genetic material and whatnot, but truly from a structure standpoint, Chargaff, he discovered the base pairing rules in the 1940s. And he discovered that basically there's a 1-to-1 ratio, okay, kind of a lock and key idea, similar to enzymes and substrates as far as things fitting together, that he looked at different species DNA, and he figured out that for every adenine there was one thymine, okay, and for every guanine there was one cytosine in a strand of DNA. So it's always a 1-to-1 ratio. Adenine pairs with thymine, guanine with cytosine. Then we have Franklin, so he's working about the same time as, as Hershey and Chase. And she was using X-ray diffraction to study DNA and try to figure out its structure. And that's an image, a top-down image of a molecule of DNA. And so she was studying this. She was really close to figuring out what was going on with the structure of DNA. And her pictures were, Watson and Crick got a hold of her pictures. And after they saw those, they very quickly started to figure out the structure that we now know of as the double helix today. They wrote a paper in 1953. It was short, it's basically a page long, and published it in 1953, and they won the Nobel Prize 10 years later in 1963. Rosalind Franklin doesn't get as much credit as she should because she was really close to making this discovery herself. She died of cancer from the X-rays that she used to study DNA, and she is a very famous scientist. She just doesn't get the same credit that Watson and Crick do. So they are credited with the discovery of DNA. If you have any questions in that structure known as the double helix, if you have any questions, let me know.